Next, we are going to look at the oxidation of carbohydrates. The oxidation of carbohydrates are going to occur when the carbohydrates are in open chain form. And what's going to happen is that carbohydrates that exist as aldehydes or hydroxyketones are going to be able to be oxidized depending on the reagents that they're exposed to. One of the main reactions that we need to be familiar with in this course for the oxidation of carbohydrates is going to be the use of a copper reagent, which we know it as the Benedict's reagent. And what happens overall is that when we look closely, and in this case, we just have the illustration for when glucose gets oxidized. When you expose glucose to the Benedict's reagent, copper 2, I'm going to highlight it in terms of the functional groups. The aldehyde in glucose is going to convert to the carboxylic acid. Understand that this reagent only acts on specific aldehydes or ketones that at the next carbon they contain an OH. So that's why this reaction works specifically with carbohydrates. In this process, since the carbohydrate gets oxidized, what we see overall is that copper 2 turns into copper 1. That means that specifically the copper reagent gets reduced. And when this reaction happens, we actually see a changing color of the solution. So when we look at the image that we have on the right side of the slide, we can see that the original color of the Benedict's reagent is this blue solution. Let me change the color of my pointer. So this is the original color of the Benedict's reagent. Then, depending on the amount of sugar that we have in the solution and the sugar that gets oxidized, that blue solution may turn into green, orange, or brick red. And depending on the color that the Benedict reagent turns, then that tells us how much sugar is going to be present in the solution. Additionally, any time that you have a sugar solution and you perform on it the Benedict's reaction, understand that the term that it is utilized for any sugar that actually has a positive effect is reducing sugar. And the reason why it's called a reducing sugar is because the carbohydrate is actually donating electrons into the copper of the Benedict's reagent to change it from copper 2 to copper 1. The next reaction that we're going to see as part of this chapter is going to be the formation of phosphate esters. Phosphate esters are actually formed by placing a phosphate group in specifically hydroxy or OH groups as part of a structure. So understand that phosphate esters, specifically when it comes to carbohydrates, are going to be present in certain carbohydrates. Not all OH groups in a carbohydrate will be obtaining this phosphate group, or in other words, is going to be phosphorylated. We can see in the bottom of the slide just two examples of sugars that have received the phosphate group. On the left, we have glucose 6 phosphate, and glucose 6 phosphate in metabolism is actually the first step in which glucose, once it enters the cell and it passes through the process of what is called glycolysis, that's the first step of the reaction, taking glucose and putting a phosphate in it. Then we see another representative of sugar that has been phosphorylated. We have here fructose 6 phosphate, and we can observe how the phosphate group has been added onto the structure. The importance of these groups, as we're going to see in later chapters, is that phosphoesters are actually going to be crucial in the structure of nucleic acids. But as I mentioned before, we are going to learn all this in detail later in chapter 13. Next, we're going to talk about glycoside formation. Specifically, we learned earlier in the chapter that when we have open form of sugars, they can cyclize and make a Haworth projection that is just showing us the formation of a hemiacetal for a particular carbohydrate. Remember that when this happens, what we have is the reaction between an OH group and the carbonyl, specifically in our structure. Now, when we have glycosides is when we are actually taking our monosaccharides and we are bonding it to an additional alcohol group. So, for example, here we have alpha D glucose. And specifically, if you take alpha D glucose and you react it with an alcohol under acidic conditions, at the anomeric position, we see the addition specifically, let me just highlight the atoms, of the oxygen in the R group in the alcohol. So what we observe here is that we go and change the functional group from a hemiacetal to a full acetal. Because now, let me just highlight the atoms. We added the OCH3 group to the anomeric carbon that we had originally in the alpha D glucose. Whenever we have the formation of such bond, meaning making an acetal, that means that overall we consider these as glycosidic bonds because we have specifically a bond formed to a sugar. Now, understand that these glycosidic bonds are going to be very important in the next class of carbohydrates, which are known as disaccharides. When it comes to disaccharides, let's understand that all of them have glycosidic bonds. And the glycosidic bonds specifically came about because the OH group of one unit of carbohydrate bonded to the anomeric position of another unit of sugar. Here we have three important disaccharides, including sucrose, lactose, and maltose. Let's talk about each of them in detail, even though we have mentioned something about them previously in the videos. Let's start with sucrose. Sucrose is known as table sugar, and it is obtained from sugarcane and sugar beets. When we look at the structure of sucrose, it is actually binding together a glucose molecule and a fructose molecule. Interestingly, the bonding, the glycosidic bond between the glucose molecule and the fructose molecule is what we know as an alpha beta 1 2 glycosidic bond, meaning that the two monosaccharides, glucose and fructose, are actually bonded by the OH. I should say the OH group at the anomeric position of fructose binds to the anomeric carbon on glucose. Let me illustrate it. So the glycosidic bond, let me highlight it. This glycosidic bond, which is right here, is because the OH group, let me change the color. The OH group from fructose is bonded to the anomeric carbon on the glucose molecule. So that's the glycosidic bond between that OH group and the anomeric carbon of the other molecule. Because of that special glycosidic bond that exists in sucrose, this disaccharide cannot form an open chain. And since it cannot form an open chain, it cannot be oxidized via the Benedict's reagent. So specifically, sucrose is a non-reducing sugar. It cannot reduce the copper 2 in the Benedict's reagent to copper 1. Next, let's look at lactose. When it comes to lactose, lactose is going to be a disaccharide that is found in milk and milk products. As we mentioned before, glucose is converted to galactose in order for the mammary glands to be producing lactose. Specifically, the glycosidic bond that is present between the monosaccharides, galactose and glucose, is going to be a beta 1,4 glycosidic bond. We can see it here. It is interesting to know that lactose, different from sucrose, can form an open chain. And because it can form an open chain, it can be oxidized with the Benedict's reagent. So lactose specifically is a reducing sugar. Lastly, we have maltose. Maltose is a disaccharide, also known as malt sugar, that it is composed of two glucose molecules. When it comes to the glycosidic bond in maltose, what we observe is the alpha 1,4 glycosidic bond, and we can see it right here. And in this 1,4 glycosidic bond, okay, it is known that maltose, specifically, it can actually form an open chain. So that means that it can oxidize, or the sugar can be oxidized with the Benedict's reagent, meaning that it actually can reduce the copper 2 in the Benedict's to copper 1. So maltose, just like lactose, is also known as a reducing sugar.